Hi, my name is Sohail. Today I am going to discuss the part 1 of eukaryotic transcription. The main points of the lesson are initiation of transcription, RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. The basic mechanism of transcription by RNA polymerase is similar in all systems. Bacteria have single RNA polymerase, while eukaryotes they have 3 RNA polymerases, that is RNA polymerase 1 transcribes ribosomal RNA, RNA polymerase 2 transcribes messenger RNA and RNA polymerase 3 transcribes transfer RNA. Together these 3 enzymes accomplish all transcription in eukaryotic cells. Eukaryotes they have membrane bound nucleus and organelles. Genes are bound in a nucleus and a eukaryotic cell must be able to transport its messenger RNA from nucleus to the cytoplasm and must protect its mRNA from degrading before it is translated. What is transcription? Transcription is the synthesis of mRNA from DNA. So, mRNA synthesized in eukaryotic cell that has a single gene. So, it is monocistronic or monogenic. But in case of prokaryotes, this is a single mRNA molecule synthesized in prokaryotes that has more than one genes. As we can see here, there is structural gene 1, structural gene 2 and structural gene 3. So, prokaryotic mRNA is polycistronic because it contains more than one gene. Prokaryotic polymerase that can bind to a DNA template strand on its own. But in eukaryotes, RNA polymerase enzyme, it requires other proteins and these proteins are called transcription factors. So, first transcription factors they bind to the promoter region of the DNA and then they help to recruit RNA polymerase 2 enzyme to form transcription initiation complex. So, transcription initiation complex it consists of transcription factors and RNA polymerase enzyme. Eukaryotes they have 3 RNA polymerases. RNA polymerase 1 transcribes ribosomal RNA except 5S ribosomal RNA. RNA polymerase 2 transcribes messenger RNA and RNA polymerase 3 transcribes transfer RNA, 5S ribosomal RNA and small nuclear RNAs. The features of eukaryotic mRNA synthesis are more complex than those of prokaryotes. In prokaryotes, a single polymerase enzyme that consists of 5 subunits, that is 2 alpha subunit, 1 beta subunit, 1 beta prime and 1 sigma subunit. But in case of eukaryote, there are 3 RNA polymerases and each RNA polymerase that consists of 10 or more than 10 subunits. So, each eukaryotic polymerase requires a distinct set of transcription factors to bring it to the DNA template strand. RNA polymerase 1 is located in the nucleolus. What is nucleolus? It is a specialized nuclear substructure in which ribosomal RNA is transcribed, processed and assembled into ribosomes. So, nucleolus is known as factory of ribosomes. Nucleolus is a region found within the nucleus that is concerned with producing and assembling the cell's ribosomes. So, ribosomes are composed of ribosomal RNA and protein. The ribosomal RNA molecules are considered as structural RNAs because they have a cellular role and they are not translated into proteins. As we can see in this slide, this is the ribosome that is attached to messenger RNA and ribosome is composed of ribosomal RNA and protein. So, ribosomal RNA are components of ribosomes and they are essential to the process of translation. What is translation? Translation is the synthesis of polypeptide chain from messenger RNA. RNA polymerase 2 is located in the nucleus and it synthesizes protein coding nuclear pre-messenger RNA. So, eukaryotic pre-messenger RNA undergo extensive processing after transcription but before translation. So, the processing of pre-messenger RNA I will discuss in part 4 of eukaryotic transcription. RNA polymerase II is responsible for transcribing majority of eukaryotic genes. RNA polymerase III is also located in the nucleus, and this enzyme transcribes variety of structural RNAs including 5S, ribosomal RNA, transfer RNA, and small nuclear pre-RNAs. As we can see in this slide, this is transfer RNA, and transfer RNA, they have a critical role in translation. Translation is a synthesis of polypeptide chain from messenger RNA. So tRNA that serve as adapter molecule between mRNA template strand and the growing polypeptide chain. So each tRNA that has an anticodon, it is a set of 3 nucleotides, and these 3 nucleotides in anticodon are guanine, adenine, and guanine nucleotides. So anticodon binds to a matching mRNA codon. Guanine is paired with cytosine, adenine is paired with uracil, and guanine is paired with cytosine. So this is base to base pairing. RNA polymerase III also transcribes small nuclear RNA. What is the function of small nuclear RNA? Small nuclear RNA, they are involved in splicing pre-messenger RNA and regulating the transcription factors. To sum up, eukaryotes, they have 3 RNA polymerases which are present in the nucleus. RNA polymerase I is present in nucleolus that transcribes ribosomal RNA. RNA polymerase II present in the nucleus that transcribes messenger RNA, and RNA polymerase III present in the nucleus that transcribes transfer RNA, 5S ribosomal RNA, and small nuclear RNA. mRNA molecule synthesized in eukaryotes that have a single gene, so it is monocistronic or monogenic, but in case of prokaryotes, mRNA is polycistronic. Each eukaryotic polymerase enzyme requires a distinct set of transcription factors. So transcription factors, first they bind to DNA promoter region, and then they recruit RNA polymerase enzyme. It forms transcription initiation complex. So transcription initiation complex, it consists of transcription factors and RNA polymerase II enzyme. But RNA polymerase enzyme is not the part of transcription factor, but it is the component of transcription initiation complex.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.